very good day, students. This lecture is on the larval forms of different animals. As we have taken the larval forms in the case of the crustaceans very particularly, and then we had taken the larval forms in the case of mollusks again in the particular lecture. Here in this particular part of the larval forms, I'll be discussing multiple phyla for their larval forms. So this lecture will be comprising of different phyla like coelenterates, the ctenophores, then we will discuss the larval forms in the case of Platyhelminthes also, the nematodes also, and the Nemertia group. So you can take this particular lecture as the miscellaneous larval forms in different phyla in the invertebrates. So first of all, we are starting with the coelenterates. So this phylum is comprising of the animals which are having a coelom, which is a dedicated coelom in their body cavities or in the body membranes. In the syllabus, you have different animals like we have gone through the Obelia and Sertularia in your BSc classes. So you can take any of the examples. But as we are discussing the larval forms in the coelenterates, so here I want to mention the larval form is called as the Planula larva. Now this particular diagram can give you an idea that how the Planula larva is formed from the zygote. Now students, as we know that in the coelenterates we have a particular colony of the animal. So if we go with this particular diagram, here it is showing you the basic colony form. Now this colony form, it is giving rise to special free living structures which are called as the medusae. So as in the case of Obelia also, we have the medusae in the life cycle, and these medusae, they can be male or female, and they are producing eggs and the sperms. Now these eggs and the sperms, they are free in the environment. These are free in the water, and they are having the fertilization process. So after the external fertilization, we have the zygote formed, and this zygote is giving rise to the Planula larva, which is actually a free swimming form. And if you go by the structure of this particular larva, it is flattened, it is ciliated, and it is bilateral symmetrical form, and it is present in various coelenterates, and also it is present in some of the species of the ctenophores. Now the planulae of this particular phylum, the larvae, they have got no mouth as such, and they don't have even the digestive tract, so they are unable to feed themselves. So they are not feeding structures basically. The Planula larva, they swim with the aboral end. You can see to some distant place. So they wherever they find some particular substratum, they follow it, and they get stuck to it, particularly that particular substratum. So whenever it swims and finds some good place, so it settles down and it develops a particular polyp, as you can see in the diagram. So ultimately that polyp will give rise to the whole adult colony form of the coelenterate. So students, that was the first larval form in this particular lecture, which is the Planula larva of the coelenterates. Now students, after the coelenterates, now we have another you can say minor phyla, which is called as the Chinofura. So ctenophores, basically they are free living forms, and I will show you one of a particular ctenophore in the diagram. So in this particular diagram you can see a particular ctenophore, and it is very well characterized like the comb jellies. So these are commonly called as the comb jellies, and they are present again in water, and they are showing a particular characteristics of bioluminescence, as you can see in the diagram. So these ctenophores, they have got a particular larval form which is called as the cediped larva. Now students, if we talk about the cediped larva, uh, it is very comparable to a planula, as it is also a free living form. So it is a free swimming larva, and this larval stage, it resembles the adults of the cediped ctenophores. So as we have seen in the previous diagram, the adult cediped ctenophore, so this particular larva is quite resembling that particular adult form. Uh, if I talk more about this particular larva, it is having a body that may be slightly compressed. So it is a dorsoventrally compressed body, and there are two retractable tentacles present, as we have seen in the case of the adult. So this form of the larvae is ovoid in shape, or you can say somewhat spherical in shape, with the two retractable tentacles. So they are also free swimming larvae of the ctenophores. Now if I take the next larval form into account after the cediped larva of ctenophores, we have got the phylum Platyhelminthes. Now students, we know that platy refers to the flat, and the helminths, they are the worms. So these are the flat worms, and if I talk about the larval forms in the Platyhelminthes phylum, the major three forms are the Muler larva, the Giotti larva, and the Catos larva. So these are the three larval forms, and we'll be taking these three forms one by one. So first of all, I must give you the structure and the type of the very first larva, which is the Muller's larva. So students, this particular diagram is giving you an overall structure of the Muller's larva. So Muller's larva is also called as the Muleria. So it is Mulleria. So malaria or Muller's larva is a larval form of some of the polyclads. So Polycladida group is having the Muller's larva. So actually these are the free living marine flatworms. So if I talk about these free living marine flatworms or Polycladida group of the Platyhelminthes, these are having the Muller's larva. Now if I talk about the structure of this Muller's larva, it has got 8-fold symmetry, and it's somewhat like the ctenophores. So if I go with the structure, it is very comparable to the ctenophores. Now the Muller's larva, it is ciliated, as you can see in the diagram also. It has got several paired and the unpaired lobes, as you can see on the lower side of the larva. Now the cilia on the lobes, they are longer than the cilia of the rest of the body. So as we have caudal tuft in the case of the Paramecium, it is on the caudal side, and the cilia, they are longer on the caudal part. So it is also present that over here that the cilia on the lobes, they are longer than the body cilia. Now at the anterior and the posterior ends of the larva, there are tufts of longer cilia, which are called as the apical tuft and the caudal tuft, as we have previously discussed right now. So the apical tuft, it originates from the apical organ. So it is on the top. Uh, the apical organ, actually it is a sensory structure which is having some function with the central nervous system of this particular larva. So as you can see in the diagram also, we have got the apical tuft on the top, and we have got the caudal tufts on the lower side of the lobes. So students, that was the Muller's larva in the case of the polyclades or the Polycladida group in the Platyhelminthes group. Now students, before going further to the second larval form in the Platyhelminthes, I must give you some idea about the two major groups of the uh, Polycladida group. Now, if I talk about you can say the flatworms. So according to the lifestyle of the flatworms, they can be classified either into the free living forms. This is the first category, the free living forms, and the second is the strictly parasitic flatworms. So these are the two basic categories based upon the, you can say, the lifestyle of the flatworms. Now, if I talk of the free living flatworms, I'm not going for the parasitic flatworms right now. So if we talk about the free living flatworms, they are directly developing into the adult forms. And if I talk about the other form, which is the parasitic form, if I talk about the polyclads, they are having the indirect development with the free living larval stages. So if I talk about the Polycladida group, as we have taken the case of the Muller's larva, it can be, the Polydida group can be divided into two suborders. Now these two suborders, they are the Acotylia and the Cotylia group. Now what is the basic difference? Cotyl refers to the sucker. So if the sucker is present, so it will be the Cotylia grouped animal, and if the sucker is absent, then it will be in the suborder Acotylia. That means that Platyhelminthes can be free living, they can be parasitic. The free living forms, they are developing directly, but if we talk about the parasitic Polycladida group, then it is having two suborders, Cotylia and Acotylia. Now, if I talk about the Muller's larva, almost all the cotylians and some of the acotylian also, they are having this Muller's larval form in their life cycle. So as we have taken the two suborders in Polycladida as cotylians and acotylians, so both cotylians and acotylians may have this Muller's larva in their life cycle. Now students, after the Muller's larva, here you can see in the diagram, if you see the A diagram, this is a Muller's larva of the cotylian subgroup, and if you take the B diagram into account, this is the acotylian Muller's larva. So that means this particular Muller's larva can be present in both the you can say orders, the Cotylia and the Acotylia group. Now students, after the Muller's larva, we have the second type of the larval form in the Platyhelminthes, and this is called as the Goethe's larva. Now students, if we take the third, the C diagram into account, this is for the Goethe's larva. And this larval form, it is only and only present in the acotylians. So this is an important thing to note. So only the acotylians, they are showing the developing juveniles, which are called as the Goethe's larva. These are having 12 eyes. So this is a very important feature of the Goethe's larva. So it is having 4 to 12 eyes, and it is having 4 lobes also. If you see the diagram, here you can see the 4 lobes posteriorly on the hind end. This particular larval form is only and only present in the acotylians. So this is a very comparable structure to Muller's larva as per the structure is concerned, but here the lobes, they are 4 in number. Now students, if we talk about the third larval stage in the Platyhelminthes group, so it is the cattle's larva. So this particular cattle's larva, actually it is again dorsoventrally flattened in structure, as you can see in the diagram. So it is a flattened kind of a larva from the center, and actually it has got 8 lobes. So 4 are present, you can see on the periphery, and 4 are present in the center. So it has got the 8 lobes. And if I talk about the eyes of this particular larva, it has got 12 eyes. So Cato's larva is a 12-eyed larval form, and previously some scientists, they have described this particular type of the larva as the intracapsular larva, but later on it was actually hatching as a bi-eyed or the 12-eyed larval form, which is called as the Cato's larva. So this is a dorsoventrally flattened 12-eyed larval form which is present in the Polyglidida group of the Platyhelminthes. And one thing is worth mentioning, that the Cato's larva is also present in the Acotylia group, as it was the Goethe's larva. Now students, if we go further of the Muler, Goethe's and Cato's larva, now we have some of the larval forms which we have taken in the graduation classes also. So we have gone through the Miracidium larva, as in the case of the flatworms or the Fasciola hepatica. So this is a Miracidium larva of Fasciola. So here you can see the major parts of this particular larval form, and these are the plates which are present on the Miracidium larva, and it is also, they are protective in nature. And here you can see the presence of the cilia, which are also present around the whole body of the Miracidium, and these are helping in the swimming motions of this particular larval form. So if I go for the important structures which are present in this larval form, these are highly specialized forms, as they have got a brain to find out the Limnia snail, as that is a secondary host for the Fasciola hepatica. The next very important thing are the eye spots which are present in the Miracidium. The next important thing is the apical gland present on the tip of this particular larval form, as this gland will secrete some of the you can say the penetration kind of the fluid, which will dissolve the shell of the snail. So that will help in the penetration of this Miracidium larva into the secondary host or the Limnia snail. So Miracidium has got these important things to reach out to the secondary host to complete the life cycle. So if I talk about the structure, the internal structure of this particular larval forms, you can have the flame cells, you have got the muscular layer over it, you have got the germ balls for the formation of the second larval stage, which is the Sporocyst larva, and this Miracidium's germ balls will give rise to the next forms, which are the Sporocyst larvae. So at the very end you have got the sub epithelium, you have the excretory pores over here, as we have got the flame cells as the excretory cells in the Miracidium larva. So this is the basic structure of this particular larval form, which is present in the Fasciola hepatica in the Platyhelminth group. Now students, this particular diagram is giving you the structure of the Sporocyst larva, or the second larval form in the case of the Fasciola hepatica. As the previous form, or the Miracidium, it enters the secondary host or the Limnia snail, that will give rise to the Sporocyst larvae inside that particular snail. Now this Sporocyst larva is having all the features of an adult form. As you can see, or the you can say on the outer side you have the cuticle, So this is the outermost layer. You have the muscular layers, you have got the flame cells, you have got the protonephridial openings for the excretion, you have got some of the germ cells which will be producing the next larval forms or the third larval forms, and you can very well see the presence of the next larval forms inside it. So on the left side I'll be demarcating, these are the, you can say, young Radial larvae which are present inside the body of the Sporocyst. So Radia are the third larval forms which will develop from the Sporocyst larva. So here you have the oldest Radial larvae, and these Radial larvae will be coming out of the Sporocyst when they are fully formed, and the next form that will be taking up is the Radia larva as the third larval form in the case of the Fasciola hepatica. Now here students, you can see the third larval stage of the Fasciola hepatica. So this is the Radia larva. These larval stages, they are having a characteristic feature of you can say the presence of the lappets. So this is the first lappet, this is the second lappet, which are present for you can say the movements inside the body of the snail. So they are helping in the movement of this Radial larva inside the snail. And the second thing which is very particular about this larval form is the presence of the collar anteriorly. So if I talk about the structure of this Radial larva, it has got a pharynx, it has got the pharyngeal glands, it has got a gut. So that means it is starting to feed on the tissues of the snail. So it will give rise to the next forms of the larvae, which are called as the circadia larvae. So you can see two circadia larvae which are present inside the body of this Radia, and this Radia larva has got a birth pore. So whenever the circadia larvae, they are fully developed inside the body of the Radia, they will come out of this birth pore, and as you can see, they have got a tail. So Radial larva will be forming the circadia larvae. Now students, this is the fourth larval stage of the Fasciola hepatica, which is called as the circadia larva. And now this larva has emerged out of the Radia, and it is present now in the snail tissues. So as it has got a tail, so it will come out of the snail. As if I go to the Miracidium larva, this is the stage which has entered into the snail. And the circadia larva, the present larval form, the fourth larval form, it will go out of the snail. So if you see the diagram on the left side, here you can see the first larval form was the Miracidium. It entered into the snail and it developed the second larval form, which was the Sporocyst larva. Sporocyst larva, it developed into the Radia larva. That was the third form, the Radial larvae. And Radial larvae, it developed into the circadia larvae. So these fourth larval stages, or the circadia larvae, they come out of the snail, and now they are free in the surrounding medium or water. So as they have got a tail, so with the help of the tail, they come out of the pond and they stick to some kind of leaf which are present around the pond. So if we see the structure of this particular larva, you have got an oral sucker on the top. So it is a very comparable structure to the adult form. It has got a muscular pharynx, it has got an esophagus, it has got a gut, these have got flame cells for excretion, and very important, they have got a cystogenous gland cells. So actually these cystogenous gland cells will form a cyst wall around this particular body of the circadia larva. So when these larval forms, they move out of the pond and they stick to some kind of leaf which are present on the outer side of the pond, these cystogenous glands will secrete some material or proteinaceous material which will form a cyst wall around this larval form, and the fifth larval form, the last larval form of the fasciola, will be developed, which is called as a meta-circaria larva. Now students, when the circaria larva is coming out, it will be secreting a cyst wall around it. As you can see in this particular diagram, this is the cyst wall, and when a cyst wall is secreted on the body of the circaria larva, the tail is lost and the whole body is encysted, and now it forms the fifth larval stage, which is called as the meta-circaria larva, and this is the final stage of the Platyhelminthes phylum as per the life cycle is concerned. So here you can see the presence of the oral sucker, the pharynx, the esophagus, the intestine. This is the ventral sucker, which is the acetabulum. Then you have the excretory canals. You have got the flame cells for excretion, and this is the presence of the excretory vesicle, which is also present in the adult form. So this metacercaria larva is very comparable to the adult form of the Fasciola hepatica. So students, these were the 5 forms of the larvae in the case of the Fasciola hepatica. First was the Miracidium, then the Sporocyst, then we have the Radia, then we have the Circaria, and then we have the Metacercaria larva. Now students, after Platyhelminthes, now we are with the nematodes. So these are the roundworms, and they are also called as the Aschelminthes group. So in this particular group of the animals, the nematodes, we have got 2 major larval forms. So the first larval form of the nematodes is the dors larva. Now if I talk about the dors, the literal meaning of the dor, actually this is the meaning is enduring, which can endure or which can cope up with the harsh environmental conditions. So students, this particular form is present in the C. elegans or the Caenorhabditis elegans animal, and if I talk about the life cycle of the C. elegans, actually it is molting uh, in 4 stages. So it has got 4 larval stages uh, if I talk about the C. elegans. So if I have to depict these larval forms in the C. elegans, that will be L1, L2, L3 and L4. So this particular animal can have L1, 2, 3 and 4 larval stages. Now whenever the L4 stage is achieved, the animal it molds to the reproductive adult final stage. So after L4, the final adult is formed. However, if the environment is unfavorable, suppose we have the L1 stage and now the conditions are not favorable to form the L2. So what will happen, that the L1 or the first larval stage, the L1 will divert itself to the formation of the dors larva. So what will happen, that L1 will form the dors larva and it will not hatch into the L2 form. So what will happen, that the dors larva is capable to cope up with the environmental harsh conditions. Now if you go by this particular diagram, so here we have the L1 larval form. So this is the first larval stage of the Caenorhabditis elegans. Now if we have the high population density over here, or we have you can some kind of stress or some kind of you can say lower food availability due to high population density, what will happen, that it will be going for the formation of the dors larva. it will not be forming the L2 stage or L3 stage. No, it will be forming the dors larval stage to cope up with the harsh conditions or low food availability. Now, if we take the second condition into account, what happens that if we have low population density, so we have got more of the food, so then it will be going for the L2, 3, 4, and finally the reproductive adult stage of the C. elegans. So one thing I want to mention over here that if the population is high, the L1 may achieve to some extent to the L2 stage. So it can go via L1 to L2, then L2 can make the dors or the dors larva. So L1 or L2 they may go for the dor larval stages, and if we have got the low population density, L1 can give rise to L2, then L3, then L4, and then the adult stage. So that's why this dors larva is very important to cope up with the harsh conditions in the nematodes. Now students, after the dors larva, we have got the second larval form, which is also present in the nematodes, and this is called as the Microfilaria form. Now if I talk about this particular form, it is an early stage in the life cycle of certain parasite nematodes. In these species, the adults they are living in a tissue of the, or you can say the circulatory system of the vertebrates. Now here you can see the larval form, which is the Microfilaria. Now it is present in the lymph glands of particular animal. So as they are parasite, or as these are the parasitic forms, so Microfilaria they are also parasitic. These are present in the circulatory system or the lymph nodes of the glands or of the animals. Now if we talk about the adult forms of the Microfilaria, actually the adult forms they are living in a particular tissue of the hosts, or they may be present in the circulatory system of the vertebrates. So the adult forms which are present in the tissues or the circulatory system of the animals, they are releasing the Microfilariae or the larval forms into the bloodstream of the host. So these larval forms they are in the bloodstream, and they are taken up by the blood feeding arthropod vectors also. So they may be you can say transferred from one animal to the other animal with the vectors. So in the intermediate host, if we have a vector as an intermediate host, the Microfilariae they may develop into the infective larval stages that can be transmitted to a new vertebrate host. So as in the case of the Plasmodium also. So students, the Microfilariae they are present in the bloodstream, and they are given out by the adults which are also present in the host. So in this particular slide they are present in the lymph gland of the vertebrate host. Now students, after the nematodes we have got the next group, which is called as the Nemertea or nemertines. So actually Nemertea should not be confused with the nematodes, as this is a different group, and it is consisting of the ribbon worms. So not to be confused with the roundworm phylum or the nematode group. So in the Nemertea group we have got two major types of the larval stages. The first form is the Palladium larva, and the second form is the dors larva. So first of all I'll be starting with the Palladium form. Now students, this is the first form of the Nemertea, which is called as a Palladium larva. Now this Palladium, it is having a developing juvenile inside it, as you can see in the diagram. It has got a developing juvenile which has a gut which lies across the larval body. So in the very center you can see the developing juvenile which is having a particular gut. It has got a mouth also between the ear flaps. So as you can see, it has got a mouth in the center, but there is no anus as such in that particular juvenile. Now a small number of imaginal discs are present inside this larval form, which will form that particular juvenile. The larval epidermis, it helps in the feeding and dispersal of the juvenile, and this is a very important function of the epidermis of the larval form. So you must be very clear about this form, that if you see the outer structure of the palladium, this is the larval form, and if you see the juvenile which is developing inside it, actually that will form the adult form. So actually these can be considered as two forms. One is the larval form, which is the outermost larval form, and you have a developing juvenile inside it. Now when the juvenile it is fully formed, the juvenile it bursts out of the larval body. So it comes out by bursting the larval body, and usually it eats it during this kind of a metamorphosis. So the juvenile which is present inside the palladium larva, when it becomes fully mature, it comes out by bursting the larval body, and then it will eat it. So the juvenile it usually eats the remains of the larva when it emerges out. So this is the story of the juvenile and the palladium larva. So one thing which I want to mention over here, it is the function of the larval epidermis. So if you take the larval epidermis into account, which is the outermost layer of the larval form, it is helping in feeding and the dispersal of the juvenile. So these are the two important functions of the larval epidermis. So this is the second diagram for the palladium larva, and here you can very well see the presence of the developing juvenile inside it. So it has got an apical tuft also, it has got the episphere or the epidermis, and it has got two lateral lobes on the lower side. Now students, we have got the second form inside the Nemertea group, and this is called as the dors larva. So according to the recent researches or recent you can say findings, the dors larva is thought to have evolved from a pelagic palladium larval form. So you can see that the dors larva is formed from the palladium, or the you can say modification of the palladium larva. Now the general course of the development has been seen to be very similar to that of the palladium. Now if you take the formation of the dors larva into account, it is very similar to the palladium larva. So here the juvenile it forms from the imaginal discs under the larval epidermis, as we have seen in the palladium also. So here also the juvenile develops from the imaginal discs which are present inside the larval form. Now during the metamorphosis process, the larval epidermis is ingested by the juvenile, thus displaying a very similar behavior to the palladium larva. So in the palladium also we have seen that juvenile when it emerges out, it will eat the larval remains. So same is the case in the dors larva. But in contrast to the palladium, there is some dissimilarity also. The function of the larval epidermis of the dors, it is shifted from the feeding and dispersal to the direct food supply. So this is a very important function. So if we take the function of the larval epidermis in the case of palladium, it was feeding and dispersal of the juvenile. And here in the dacor, the function of the larval epidermis is to give the direct food supply to the developing juvenile. So this is a dissimilarity kind of a thing. Otherwise, both the larval forms, they are very similar in the development aspect. So students, with this we are finishing with the miscellaneous larval forms of the coelenterates as the Planula larva. In the Chinofura group, we have gone with the cediped kind of the larva. If we go with the Platyhelminthes, we have got the Muler larvae, the Goetes larva, the Catos larva, and then we have got the Miracidium, the Sporocyst, the Radia, the circadia, the metacircaria larvae. And if we go by the nematodes, we have got the dors larva, we got the Microfilaria, and if we go with the Nemertine group, we have got the Palladium and the dors larva. So with this we finish with this particular lecture. Thank you so much.